So welcome back again. Let's take a look at the concept check questions so that we can move on and talk about the actual way in which meiosis occurs. So in the cell shown in slide 23, how many individual DNA molecules or potential chromosomes are there? What is the haploid number, and is a set of chromosomes diploid or haploid? So this is the slide that we're talking about. So if we look at this, how many potential chromosomes are there? Well, remember, each sister chromatid represents a potential chromosome and also represents a single DNA molecule. So we've got 6 chromosomes, but we've got 2 sister chromatids in each chromosome. So it's going to be 6 times 2, that's 12. So there are 12 DNA molecules or 12 potential chromosomes in here. Then the second part of the question asks, what is the haploid number? Well, the diploid number is 6. That's 2n. We know the haploid number is n, so that must be half of 6. So that would be 3. So the haploid number is 3. And then is a set of chromosomes diploid or haploid? And there we have to remember our definitions of diploid and haploid again. Remember, diploid means 2 complete sets of chromosomes. Haploid means 1 complete set of chromosomes. So a set of chromosomes must be haploid. It's 1 complete set of chromosomes or 1 set of chromosomes. That's the haploid number of chromosomes. The diploid number, remember, is 2 complete sets of chromosomes. In a human karyotype, how many pairs of chromosomes are present? Well, we know that humans contain 46 chromosomes. That's our diploid number. But that means there must be half that number pairs of chromosomes, because each of those 46 chromosomes has a partner. So that means there must be 23 pairs of chromosomes in a human karyotype. How many sets of chromosomes in a karyotype? Well, remember again, we're looking at a cell that is diploid. That's the only way we can see the homologous pairs of chromosomes. And so there would be 2 sets of chromosomes and the cells would be diploid by definition. Okay, let's go on and talk about how meiosis actually takes place. So as I was saying before the break, meiosis has to be, by definition or by requirement, a 2-step division process. So we talk about meiosis 1 and meiosis 2. Meiosis 1, as I emphasized before the break, is where we do actually reduce the chromosome number from diploid to haploid, from 2n to n. So we're going to be separating the homologous pairs. Then in meiosis 2, we have to remember that all of these homologous chromosomes at the beginning consist of 2 sister chromatids. So in meiosis 2, we're all about separating the 2 sister chromatids. And not surprisingly, it looks a whole lot like mitosis as a consequence. So these are the 2 stages of meiosis. And so here you can see our artist's rendition of what goes on prior to cell division. Again, because it's cell division, we're going to duplicate the chromosomes. When is this going to occur, you might ask? Well, when do we duplicate chromosomes before mitosis? And you should be going S, it's the S during interphase, because that's the same here. It's still cell division, nothing changes. So the chromosomes are duplicated during the S phase of interphase. So at the beginning of meiosis, we have duplicated chromosomes, 2 sister chromatids in each. So meiosis 1 then separates the homologous pairs. And so you can see here, change this to red, that this is what is going on. We are separating the homologous pairs of chromosomes in meiosis 1. So 1 member of each homologous pair goes this way, 1 member of each homologous pair goes this way. Then in meiosis 2, it's all about separating the sister chromatids. So 1 sister chromatid goes here, 1 sister chromatid goes here. But we've got 2 cells undergoing meiosis 2. So in the end, we wind up with 4 daughter cells. And they are not genetically identical. So immediately we start to see some differences between the cell division that we know as mitosis and the cell division that we now are identifying as meiosis. The end result is very different. Mitosis, 2 daughter cells genetically identical to each other and genetically identical to the parent cell. At the end of meiosis, we have 4 daughter cells. They are not genetically identical to each other, and they are most definitely not genetically identical to the parent cell. So immediately we start to see some differences. So remember again, meiosis 2 looks very much like mitosis, and I'll show you the stages, but we're going to spend a lot of time looking at meiosis 1, because that's where all the differences that are associated with meiosis occur. And so we'll spend a lot of time talking about meiosis 1. So let's take a look at the stages of first meiosis 1 and then the stages of meiosis 2. Now, the first thing to recognize is the names of the phases up here, prophase, metaphase, anaphase, telophase, fit into the PMAT format. Now, when we were talking about mitosis, remember we were using PPMAT because there was the prometaphase inserted between prophase and metaphase. I don't know of any freshman biology textbook that includes prometaphase in meiosis. In fact, I've only ever seen it included in a description of meiosis in an animation that was produced for a PBS special about 18, 19 years ago on multiple different ways to make a baby that did not involve sexual reproduction. So I was talking about IVF and other processes of ways of making babies other than through intercourse and sexual reproduction, normal sexual reproduction. Someone produced an animation of meiosis that did include a prometaphase in both meiosis 1 and meiosis 2. But every other textbook that I've ever looked at and every other resource that I've ever looked at typically does not include a prometaphase. So we can use PMAT here, prophase, metaphase, anaphase, telophase. Now notice that they're designated prophase 1, metaphase 1, anaphase 1, telophase 1. And you might ask why? For a very simple reason. The stages in meiosis 2 are given the same name, prophase, metaphase, anaphase, telophase. If we were just to call them prophase, metaphase, anaphase, telophase, we would then have to ask, but which are we talking about, meiosis 1 or meiosis 2? So rather than get into that, we simply call them prophase 1, metaphase 1, anaphase 1, telophase 1, indicating they are in meiosis 1, and we call them prophase 2, metaphase 2, anaphase 2, telophase 2, because they're in meiosis 2. Now, the names of the phases are not accidental. There is, in fact, a connection back to mitosis. It's all about, remember, in mitosis, what is happening with the chromosomes in the nucleus, and specifically what's happening with the individual chromosomes. In meiosis 1, not so much meiosis 2, but in meiosis 1, much of what we could describe in mitosis, we can actually describe in meiosis 1. The big difference is that rather than talking about the individual chromosomes, we simply substitute talking about the homologous pairs of chromosomes. Everything else pretty much remains the same. So if we think about prophase, and because we're doing PMAT rather than PPMAT, we can incorporate prophase and prometaphase here in terms of what sort of things are going on. So if we consider what goes on in prophase and prometaphase of mitosis, well, we know that the chromosomes condense out and become visible. They undergo that supercoiling process and become visible, and each of the chromosomes contains two sister chromatids, we know that the spindle starts to form as the centrosomes start to move apart from each other. The microtubules go out and start to form the spindle structure. Later in prophase, we know what we might call prometaphase, but here we can call late prophase. We know that the nuclear envelope breaks down, and we know that some of the microtubules, what we referred to as the kinetical microtubules, engage with the individual chromosomes via the kinetochores. That's all going on during mitosis and, prophase and prometaphase of mitosis. Those things are also occurring here in prophase of meiosis, or meiosis 1. So do the chromosomes condense out? Yes, they condense out and become visible. But it's not the individual chromosomes that condense out and become visible. It's the homologous pairs that condense out and become visible, because they are already partnered up with their homologous partners when they undergo the supercoiling and become visible. And they are held together by those synaptonemal complexes that I was referring to that form the chiasmata, singular chiasma. So here you can see a pair of homologous chromosomes being held together by two chiasmata, that's the plural, between non-sister chromatids. So there it is, non-sister chromatids form the chiasmatal or synaptonemal complex links between the two homologous chromosomes, the two homologous partners. And so when the chromosomes condense out, they condense out as their homologous pairs, held together by these links, the synaptonemal complexes or the chiasmata. The spindle is forming, nothing new there, being generated by the migrating centrosomes as they move apart from each other. The nuclear envelope eventually breaks down, nothing new there. Eventually, microtubules from the spindle start to engage with the chromosomes via the kinetochores, nothing new there. But it's not with the individual sister chromatids, it's with the individual homologous partners. So microtubules from one end of the spindle connect with the chromosome, one homologous partner. Microtubules from the other end of the spindle connect with the other partner, so that each homologous chromosome is connected to one end of the spindle, and each of the partners is connected to opposite ends of the spindle. So if we look at the chromosomes, once they line up on the metaphase plate, which remember is what happens in metaphase of mitosis, but it's the individual chromosomes that line up on the metaphase plate. Here in metaphase 1 of meiosis 1, it's the homologous pairs that line up on the metaphase plate. And they are guided there by the microtubules, just as the individual chromosomes were guided there in mitosis. Now, here's where it starts to get a little bit complicated, and we'll address this complication a little bit later. But notice that these chromosomes line up in different ways. These two pairs of chromosomes happen to have lined up with the blue chromosome on the bottom and the red chromosome on the top. But this one's gone about it the other way, put the blue at the top and the red at the bottom. And in fact, each of these pair of chromosomes could have lined up either way. Red at the top, blue at the bottom is just as legit as blue at the top, red at the bottom. And each of these chromosomes could have lined up either way. And what one pair of chromosomes does is completely independent of what the other pairs of chromosomes do. So what's important to recognize here, and I'm going to bring on, where am I going to put that? I think I'll put that up here. Each pair of chromosomes lines up randomly, so it could be one way or the other, and So we've had the PMAT there for a while. I'm going to take the PMAT out now so that I can have the space. Maybe I'll put it in. I'll put it down here because I may not need this space. So again, it's PMAT, prophase, metaphase, anaphase, telophase. So the important thing that we need to remember here, and we'll talk about the significance of this a little bit later in the last part of the chapter, is that each of these pairs of chromosomes lines up randomly, so it could be red at the top, blue at the bottom, or the other way around. Each orientation is completely legitimate and possible. And what one pair of chromosomes does is completely independent of what the other pairs of chromosomes do. And that has significance, as you'll see. Then in anaphase of mitosis, the individual chromosomes separate and start migrating to opposite ends of poles as the centromeres, break and allow the sister chromatids to separate. Here, it's the homologous pairs that separate. So the synaptonemal complexes, the chiasmata, break and allow the individual chromosomes, the individual members of the pairs, to migrate to opposite ends of the spindle. So one member of each pair goes in this direction, one member of each pair goes in this direction. So we have one member of each pair arriving at each end of the spindle. And what that has effectively done is reduce the number of chromosomes by half. So this is where the actual reduction in chromosome numbers is taking place, when the homologous pairs separate because the chiasmata break and allow them to separate and migrate to opposite ends of the pole, opposite ends of the spindle. Then, depending on whether we're making eggs or sperm, a new nucleus will form and cytokinesis will accompany telophase. So just as we saw in mitosis, new nuclei form up in the two daughter cells and cytokinesis occurs in parallel with telophase. So we see that happening here. Now, if we were creating sperm, this would most definitely be happening. We would be dividing the cells into two new daughter cells. If we're making eggs, cytokinesis does not happen. New nuclei form, but cytokinesis does not happen. So let me put that in here. So that's an important observation. What happens next also depends on whether we're talking about testicles or ovaries, as far as animals are concerned, and certainly as far as humans are concerned. Because the egg cell does not complete meiosis 2. In fact, it may not even complete meiosis 1. It's going to get held at some point in the middle of meiosis somewhere. Somewhere around the end of meiosis 1, the beginning of meiosis 2 is typically where the egg cells are held. And they actually will not complete meiosis until they're fertilized by a sperm cell. Let me say that again. If we're talking sperm, cytokinesis is going to occur and the cells will go on and complete meiosis too. But if we're talking about making egg cells, number one, cytokinesis is not going to occur. And the cell is not going to complete meiosis too. It's not going to complete meiosis. And it won't finish meiosis until it gets fertilized by a sperm cell. That also has consequences. It's something that we do need to talk about a little bit. We will touch on it a little bit at the end of this chapter, but it will become more of an issue when we talk about inheritance patterns in chapters 14 and very specifically in chapter 15 as far as this is concerned. But I'll touch on it a little bit before we finish up today. So we are now at the end of meiosis 1. Let's assume that we have two daughter cells, each containing a haploid number of chromosomes at this point. We've effectively reduced the number of chromosomes from diploid to haploid. We've got two daughter cells, each containing a haploid set of chromosomes. But they still contain two sister chromatids. Each chromosome still has two sister chromatids. We need to separate them. So we undergo a process or a form of cell division that looks very much like mitosis. To all intents and purposes, it is mitosis. So again, the spindle starts to form, the chromosomes condense and become visible, and this time it is individual chromosomes. The nuclear envelope degrades. Kinetochore microtubules now connect with the individual chromosomes via their kinetochore structures. Each sister chromatid is connected to one end of the spindle. Its partner is connected to the other end. They eventually line up on the metaphase plate, and notice again, it's the individual chromosomes. Again, sounding very much like mitosis. In anaphase 2, the centromeres rupture, allowing the sister chromatids to migrate to opposite ends of the pole. Nuclei form up, and in the end, we have four daughter cells, each containing a haploid number of chromosomes. But this looks very much like mitosis. And in fact, for all intents and purposes, you could call it mitosis, but there are some subtle differences that we will address when we look at our concept check question, because it actually addresses this. So this is meiosis 2, looks very much like mitosis. Back here in meiosis 1, all the different steps of cell that are not identical to mitosis are occurring, but remember, there is some connection back to mitosis. We see it reflected in the names of the stages, and we see it in terms of some of the things that are happening. The big difference, of course, is when we talk about what the chromosomes are up to, rather than talking about individual chromosomes, we have to remember that we're talking about pairs of chromosomes. So whatever we talked about in mitosis, concerning individual chromosomes, we talk about here in meiosis 1, referring to the homologous pairs rather than individual chromosomes. Otherwise, everything remains very much the same. So those are the two stages of meiosis, meiosis 1 and meiosis 2, and what goes on in each of the two stages. So let's talk about what happens when the non-sister chromatids actually link up. The synaptonemal complexes or the chiasmata. It actually turns out to be a whole lot more interesting than that. The proteins that are involved in holding them together are called cohesins. And it just creates a cohesive link between the two non-sister chromatids. When we take a look inside the synaptonemal complex, what we see are that there is not just a protein complex connection, but there can be a direct DNA to DNA connection between the two non-sister chromatids, so that not only do you have proteins holding them together, you actually have links to the two DNA molecules, so that there is a cross-link, what we refer to as a crossover, between the two DNA molecules that represent the two non-sister chromatids. And that turns out to be important because when the chiasmata break, those cross-links between the two DNA molecules may remain intact, which means that when the two non-sister chromatids separate from each other again, when the two homologous partners separate from each other, there's an actual physical exchange of DNA between the two non-sister chromatids as a consequence of the crossover. And this exchange of DNA is actually really important because it is a potential source of genetic variation, which we'll discuss in a little bit more detail after we take our final break and get into the last section of the chapter. But these are the links that are holding the homologous pairs together during that initial part of meiosis 1, during prophase 1 and metaphase 1. These links break in anaphase 1 to allow the homologous pairs to separate and migrate to opposite ends of the chromosome. But as a consequence of these potential physical connections between the actual DNA molecules, there is a very real potential for a direct exchange of DNA between the non-sister chromatids, and that has consequences. So when we start thinking about how do meiosis and mitosis compare, are there some differences? Are there some similarities? Well, we can say with absolute certainty there are some distinct differences, but there are some somewhat similar events in terms of formation of the spindle from the centromeres, loss of the nuclear envelope, kinetochore microtubules attaching to kinetochores, separation of sister chromatids in meiosis 2 at least. But in terms of what goes on, in terms of looking at cells that are undergoing meiosis and being able to identify them from cells undergoing mitosis, it depends on whether you're looking at meiosis 1 or you're looking at meiosis 2. Because remember, again, meiosis 2 looks just like mitosis. So in general, you probably would struggle to differentiate a cell that's undergoing meiosis 2 from a cell that's undergoing mitosis. There are some ways of identifying it, but it would be a tough decision to distinguish. But in terms of meiosis 1, yeah, it's pretty easy because there's going to be those synaptonemal complexes, a process called synapsis, and there's going to be the cross connections, the crossing over, and that you're going to see at the beginning of meiosis 1 during prophase 1. And you can identify that in the microscope because remember the chromosomes are visible. So you can actually identify this. Then when we're looking at metaphase 1, you're actually going to see the homologous pairs lining up on the metaphase plate, what are referred to as tetrads. So another way of referring to the homologous pairs, certainly as you see them in the microscope, is as a tetrad. So of course, tetra means 4. So a tetrad simply refers to the fact that there are 4 chromatids, the 2 sister chromatids of each chromosome, making up 4 chromatids within this structure. And so that's what the origin of the term tetrad. And that's what lines up on the metaphase plate. Now, you can't see the random arrangement of that lineup, but you can certainly see them lining up on the metaphase plate in a light microscope. If we are thinking in terms of separation, if we look at the two anaphases, well, in anaphase 1, we know it's the homologous pairs that separate. In anaphase 2, we know that it is the sister chromatids that separate. So you can actually differentiate anaphase 1 from anaphase 2 at the light microscope level, because you can pick the difference between the homologous pair separating, because the chromosomes still consist of two sister chromatids, from the sister chromatid separating, because you can identify the individual chromosomes once the sister chromatids separate and become daughter chromosomes in their own right. That's typically visible in the cells. So it is possible to separate or to identify anaphase 1 and anaphase 2 individually in a light microscope situation. So basically in this diagram, you have laid out what goes on in mitosis versus what goes on in meiosis. And basically it's a comparison of mitosis with meiosis 1 and to some degree meiosis 2, because we know that meiosis 2 looks just like mitosis, but if you're comparing meiosis with mitosis, to see the real differences, it's comparing meiosis 1 with mitosis. And again, in mitosis, it's all about what the individual chromosomes do. In meiosis 1, it's all about what the pairs of chromosomes do. Of course, in meiosis 2, again, it's all about what the individual chromosomes do, because it looks just like mitosis. So, time for our last break in this presentation. We still have one more concept check right at the very end, as we typically do, but this is our last break for a concept check. So, here's the question. Compare and contrast chromosomes of a cell in metaphase of mitosis with those of a cell in metaphase 2 of meiosis. So here we're looking at mitosis and the stage of meiosis that looks just like mitosis, or at least at first glance does. We'll see you on the other side of the break with the answer.